Good afternoon, everyone. Thank you all for joining us for day 2 of the 14th Annual Vermont Organics Recycling Summit, hosted by the Composting Association of Vermont and the Vermont Agency of Natural Resources, Department of Environmental Conservation. I'm Natasha Duarte, the Director of the Composting Association of Vermont. I'd like to just take a minute to thank ANR for their continued partnership, especially for their support and flexibility as we navigated first the postponement and then the new format of VORS this year. I'd also like to thank my board of directors for their input and support with the changes caused by the pandemic and in organizing VORS as a webinar series instead of an in-person event. Many thanks as well to our VORS sponsors who also hung in with us. Many signed up for the in-person event sponsorship and were really understanding as we tried to figure out how to still hold the summit this year in a virtual format. And that's Addison County Solid Waste Management District, AgriLab Technologies, Community Bank, Eco Products, Nature Cycle, Rich Earth Institute, Simplicity Engineering Solutions, the Trade Council of the Ministry of Foreign Affairs of Denmark, and Vermont Natural Ag Products. Huge thanks to them. Just a little promo for the rest of this week's program. We have a lot of great webinars lined up for you, even though it's a slimmed down schedule compared to the in-person event. So we hope many of you will be able to join us for multiple webinars this week. And so moving on to our session today, I'd like to introduce Deb Nair, who will be the facilitator for our webinar. Deb is a soil ecologist in the Department of Plant and Soil Science at UVM. During the past 10 years, her research has included pilot studies on biological communities in compost and their role in disease suppression. Currently, she has ongoing farm trials testing compost-based products for management of bottom rot disease of lettuce caused by globally distributed soil-borne fungal pathogens. She views nature as a model and is a question-driven researcher using population and community ecology approaches. And so I'm going to stop sharing my screen now and pass it over to Deb, who will take over for the session.
Thanks for that introduction, Natasha, and hope everybody's doing well today. Let's see, can you see my video?
Yes.
Okay,
thanks.
Awesome. So, so here's the kind of the structure that we'll use for today's session. I'll start out by talking a little bit about pathogens, antibiotics, and antibiotic resistance. And then I'll be followed by Ned Beecher, who will look at implications for compost, focusing on PFAS. And he will be followed by Andrew Carpenter of Northern Tilth, who will be talking about other chemical and physical contamination issues. And then I'll return for a brief discussion on microplastics. And we have plenty of time allowed for questions and discussion. All right, in the 1970s and before, biosolids were called sewage sludge, and it kind of reflects how humans interacted with them. Many coastal cities just dumped them into the oceans as a mean of disposal. But then we started realizing that there actually was some utility and reason why we wanted to return that organic material to the land. And so US EPA kind of partnered with USDA's Agriculture Research Service to look at the how we could use composting and the proper techniques of that composting to manage and destroy any pathogens that might occur in that biosludge. And since then, actually, we've converted that name to biosolids, which I think has a less negative connotation to it in terms of that, and thinks about it more as a resource rather than a waste. This partnership between USDA and US EPA really generated an incredible body of research that's really the linchpin of the US composting industry that persists today. And out of that, one of the major features that persist today is that we know that thermal requirements, that if we attain those will remove pathogens. And it varies, the period of time needed for that temperature varies whether we compost through a windrow method or whether it's through an aerated static pile. Illustrated on the right-hand side just kind of illustrates some of the data we have for looking at how if we attain high temperatures, the temperatures exceeding be 130 over 130 to 170 degrees Fahrenheit, where we get really good kill of coliform bacteria. You can see here on the Windrow method plot, we can see on the y-axis is the bacterial concentration, and on the x-axis is time, and we can see those populations of the coliform and specifically Salmonella decrease as that temperature reaches those levels. The period needed for the good pathogen kill is much shorter in an aerated static pile. So we've been able to, adopt these as standards that we've used in the industry over the last few decades. We've also tested this kind of approach here in Vermont, and this was a demonstration project that was funded by the Northeast Sustainable Ag and Research and Education Center, and it was a project that I was collaborating with the Highfield Center for Composting. And it occurred about a year, it was a year after a very severe epidemic of late blight on potatoes, and farmers were in a quandary. And this is the same pathogen occurs on tomatoes as well. And farmers were asking, what do I do with my diseased plants? And some folks were saying, just put them in the landfill. But this project was to demonstrate if we followed the thermal guidelines for composting, that we could actually keep all those nutrients on farm and have a complete kill of pests, including not only plant pathogens, but also weed seeds. So in this trial that we did, we had 3 carbon types that we looked at, and we used hay and a softwood and a hardwood and matched our got our 25 to 30 to 1 carbon to nitrogen ratio with optimum moisture and turning. And we used as a control kind of what I'll call kind of a farmer control, which was just a dairy manure mixed with bedding. That would be what might be pushed out of the barn at the end of a winter and into spring. You can also think of this as an equivalent of kind of cold composting. And we put into these different piles, we put in the weed seeds. We used large crabgrass seed, and then we had a pathogen that we put little discs in these bags. The bags were either solid plastic or had mesh, so that the solid ones would only allow heat transfer, and those with mesh would allow both microbes and the heat to transfer to those pests or pathogens. The time scale here was relative. They were just different. We collected these bags of the seeds and pathogens at different times through the composting process. The T1 time actually corresponds with the National Organic Program Standard, and which is recommended by the Northeast Organic Farming Association. So the point here is to focus on T1. In all of these cases, you can see that the viability of both the crabgrass seed and the pathogen declined to 0 by T1, which is the time recommended for a pathogen kill. So the bottom line is this method works as long as we're applying, having appropriate carbon to nitrogen ratio and attaining those temperatures, which may be either through forced air or by turning. Now, the one thing that's a little bit of is a little bit different here is the blue line that occurs here on the heat only. That happens to be the quote cold compost and manure. It's just illustrating some of the natural variation there. But, by the time that it reached the end of the thermophilic phase, well into the maturing phase, there was 0% viability of either the pathogen or the weed seeds. So the bottom line there, the 2 take-home messages here we can have is, one is actually we get better kill if we allow for the microbes and the heat. It's not just heat, but microbes will actually, accentuate that effect. And then secondly, we do get kill with this, even if we aren't attaining in this colder compost, not quite to recipe, but it just takes a lot longer. We can get a product in 6 months versus 2 months if we do follow the rules and regulations. So bottom line is we know how to kill pathogens. And all of this work on these guidelines originated from that work in the 1970s. Now, these kinds of experiments have been repeated for a wide variety of pathogens, and the biggest concern has been thinking about composting of manure-based products from a variety of animals. So I've organized these a bit by, the type of pathogen you can see. There's a number of bacteria that are related with food safety concerns or various originating from manures of different animals. They're all killed with this thermal requirement. Protozoa are killed. There's variety of, then on the right-hand side, these 3 columns relate to plant pathogens. The Mycota, Pythium, and Phytophthora are very well managed with compost, manure-based compost, as are other root pathogens that are fungal or nematode in nature. Now, since we're in the midst of a pandemic at this time driven by a virus, people might be curious whether or not composting will denature viruses. The fortunate thing we have with viruses is they are not saprophytes. They're not decomposing this material, and that's what composting is a controlled decomposition process. Viruses are obligate parasites, and they're not going to survive if they don't have a living host. So it's really kind of a moot point. The only concern is the things that we deal with anyway if we happen to sneeze on something, but that's a short-lived phenomenon. So not to worry about the viruses. So another area that, so we know how to kill pathogens. Now there's another big concern that we deal with that's kind of related to management of pathogens, and that is antibiotics or bacteria that have developed resistance to antibiotics, or the genes that may be resistant to antibiotics. And one thing for sure is that we have a lot of research evidence is that these are not properly managed and handled by, flushing them down the toilet and letting them go to the wastewater treatment plant. They're not going to degrade very effectively doing that. So that's why we have the big X across that to even suggest it. However, the good news is, well, maybe back up for just a minute. One thing we're concerned is just the antibiotics themselves. And let me say antibiotics are nature. That's in nature and natural. Bacteria, fungi naturally produce antibiotics for their own personal defense against other microbes they interact with in the environment. And humans have harnessed that to, for our own medical care. And, we also use those in veterinary science for livestock and animals. But there's been such a large use of antibiotics that we are developing a very strong selection pressure for resistance to develop. And once we have resistance to these antibiotics, they no longer become effective for their use in public and environmental health. So we're very concerned about the magnitude of resistance that's evolving, and then whether or not we have a means to manage this if it is in the environment. And the evidence is that compost is effective at destroying antibiotics, and I'll show you some data to support that statement. One of the challenging things about bacteria is they not only can carry genes for resistance in their chromosomes, but they also have a lot of these what we call mobile genetic elements in the cell that allows them to transfer these chunks of DNA that confer this resistance to other organisms that might not have it, or they can stack them up so they can have multiple resistances. Here's a recent study published that provides evidence to show that composting has the ability to reduce the abundance of these antibiotic resistance genes and these mobile genetic elements. On the left is a table that compares different organic waste treatments This is the result of it's a metadata analysis. They included over, I think it was about 1,300 studies in the overall thing, but you can see those they compared actually in this table. And I've highlighted the line here with composting, showing that with proper composting, there's an 84% reduction. That's the mean. And the range is 65 to 93% reduction of antibiotic resistance genes and these mobile elements. And this is highly statistically significant. And on the right-hand side is the global effect size, and we see the dot here, the very small error confidence interval or error bars compared to some of the other treatments. Now, in contrast, if we look at digestion, whether it's aerobic or anaerobic, it's a large amount of variation, and it's insignificant in terms of their ability to actually destroy these genes or elements. And on the right-hand side, we can see the interaction of temperature with that. And on the top is an illustration of the composting. On the y-axis is the reduction of these resistance genes and these mobile elements. And we see there's really no significant difference of temperature here. So what that's telling us is that it's really the microbial interactions in that compost that are destroying those genes and those mobile elements. That it's not the temperature, but it's that microbial component. In contrast, at the bottom, if we look at the anaerobic digestion, we do see a significant effect or relationship of temperature. When that is at a relatively low temperature, there's very little reduction in these the abundance of these genes or mobile elements. But if there is an increase in temperature, then there is an increase in reduction. Another study that was just published in June this year looked at a variety of genes associated with very commonly used antibiotics and looked at the decay of these genes in composting, whether these antibiotics were introduced as individual compounds or mixtures of compounds. So the T0 to T5, let's say, it's an absolute abundance on the y-axis, and on the x-axis we see treatments and time illustrated. The treatments have numbers from T0 to T5. The T0 is no antibiotic control. T1 through T4 are single antibiotics, and I've listed those below: lincomycin, chlorotetracycline, sulfamethoxazole, and ciprofloxacin. And then we see each of those through time. So there's a couple take-home messages here. Well, one other thing is the different colors just represent different resistance genes that were included. For example, on the panel A is for tetracycline resistance gene. In the middle are related to some of these mobile elements, as well as resistance to sulfonamide and quinolone and macrolides on the right. We see in all cases a general pattern with time. We see kind of a exponential decay in the relative abundance of these resistance genes. So that's good news and provides evidence that composting is reducing these. Now, another way that antibiotics can interact here is that if you have antibiotics, there is the potential to actually slow down decomposition, because it would be, say, reducing some of the microbes that you would want there to be decomposing. The only time they found that there was any delay in obtaining the heat requirements was when they had a mixture of these antibiotics. When they mixed all four of them, they had some delay at the beginning, but eventually the reduction of those resistance genes, it caught up. So all in all, whether those antibiotics are in single or combination, we saw a reduction. So I hope with these few slides that I've convinced you that we know how to deal with pathogens, and we should really be focusing our attention more on some of the other emerging pathogens. And that's what I want to start turning the table over to Ned and Andrew for, who are going to talk about PFAS, some of the persistent herbicides, and other personal care products. So I will first introduce Ned. So Ned Beecher, he was formerly the executive director of NEBRA. He's currently the special projects manager. NEBRA stands for Northeast Biosolids and Residuals Association. And they've been focusing a considerable amount of their work since January 2017 on PFAS. NEBRA is a nonprofit organization. Their mission is to advance best practices in sustainability and biosolids recycling. Ned's led a variety of projects and authored articles, papers, book chapters on biosolids management in the Northeast, Eastern Canada, and around North America. He holds a master's degree in resource management from Antioch University and a bachelor's in geology from Amherst. And he's won a couple significant awards during the last few years. He earned the New England Water Environment Association Biosolids Management Award, as well as the Elizabeth Coutone Executive Leadership Award. Take it over, Ned.
All right, hopefully you can hear me.
Yep, you're good.
Please let me know. Okay, so welcome everybody. Thank you for attending this session, and hopefully it's helpful. I'm going to move quickly through my slides, and I'll offer that these slides will be available, and if you will want to look at more details and read them more closely, they'll be available. So thank you, Deb and Andrew and Natasha, for organizing this, and to VORS and ANR for supporting this effort and outreach. So we're talking generally about recycling organic wastes, and composting is a major part of that, an important part of that in society. And we do this because it benefits society and the environment by managing materials that we need to manage. They have to be treated, tested, regulated, and recycled to get sustainability going. And we've been doing this for a long time. These are all the kinds of benefits that accrue from that. And you know these things as professionals in the field. But there are trace contaminants. There are concerns that we have with various organics, and they end up taking a lot of our time and attention. We do need to manage them carefully, but it's always good to be reminded of the benefits. What about contaminants of emerging concern? So I'll use CECs as the acronym here. Some people use, and some of the literature uses TORCs, toxic organic contaminants is another. So there's different terms for this, but CECs tends to be the predominant one. So organics have had the attention, as Deb was saying, of the concern about CECs for quite some time. Biosolids, which is the focus I tend to address mostly, have been often the most studied because of the potential for mixtures and things in biosolids. So much of the research on this has been focused on biosolids, but much of it applies still to other organics, from food waste to yard and leaf waste to whatever. It can all be very applicable to whatever you're working on in composting. The key thing that really brought the CECs to our attention at the time was the USGS research finding various trace chemicals in streams across the U.S., including personal care product chemicals and others. That led to an expansion of the amount of research on this topic of trace contaminants in the environment. There was a lot of interest in dioxins and furans back in the late 1990s and early 2000s, and that led to a very careful risk assessment by EPA with a robust database looking at whether dioxins and furans needed to be regulated, and it was determined they did not need to be given the levels in biosolids. So We have a lot of more information on microconstituents, as they're often called as well, torcs or CECs, and these are all the different kinds of materials, the PPCPs, pharmaceuticals, and personal care products that have been looked at by various research over the decades. So we'll look at a few of these and a few of the findings just to get a sense of it. Andrew will talk about persistent herbicides later on, and Deb will finish up with some discussion of microplastics. Both I and Andrew will be getting into PFAS, but generally the best method to deal with these, as I'm sure everyone agrees, is if there are chemicals in use that are of real concern for the environment or public health, then we should be phasing them out as soon as we know that. So many of these have already begun to be phased out. So concentrations of CECs in biosolids don't necessarily represent the concentrations in compost you might be dealing with, but this is sort of a big picture look at the different levels of things in biosolids, wastewater sludges. So there can be very large, this is a logarithmic scale, and so it can be very large differences in actual concentrations, and the concentration doesn't really represent risk directly. There can be high levels of something that is low risk, and then small levels of things such as the perfluorinated chemicals now called PFAS, here down at the right end of this, which even low levels are driving some concerns.
Can I interrupt you for a second, Ned?
Yes.
Your voice, it didn't cut out, but it got a lot lower in volume in the middle of that last slide.
Okay. Well, thank you for letting me know, and please speak up if it becomes hard to hear me. Much of the research on CECs is for biosolids, as I said, but one of the clear findings that Deb's presentation pointed to, and which we see in numerous studies, is that composting certainly degrades many CECs. The vast majority are broken down by the various microbes in composting and later on in the soil itself. But composting is an excellent way to degrade organic residuals, organic materials, organic compounds. So it's really one of the best possibilities, technologies really, for degrading this, and it's actually used to break down organic contamination in situ in the field, as you know. Even the worst case field application scenario, spiking pharmaceutical and personal care products in a field study in Ontario, for example, measured PPCPs in tile drainage, but at fairly low levels. There have been looks at whether these are being taken up by earthworms and such, and USGS research has certainly confirmed that. So there is concern about food chain transfer in that way. So research on that has continued. But the impacts, as we'll see in a couple of the slides here, seem to be relatively low, even for most of the pharmaceutical personal care products and CECs when you land apply them in compost or biosolids. So what counts in any of this is not the presence as much, that if there's a trace chemical in the compost or in a biosolids. What counts is the levels and what the fate and transport are, where they go, whether they get into plants, whether they get into groundwater or surface water, or whether they just stick in the soil, whether they're decomposed. So the fate and transport is critical, and then the impacts is sort of, is there an organism that is impacted? So here are a couple of findings, again, from work in Ontario. Maximum concentrations in effluents are generally below toxic thresholds, so that's in the surface water or in groundwater. Even with biosolids applications, pretty heavy levels, we're not seeing most of the PPCPs and similar trace chemicals causing any concern. What is important is to understand how the research is done. for example, there can be lab studies and greenhouse studies where you end up spiking chemicals into the samples that are being tested to see if there's leaching. And that will always overestimate what would happen in a field condition for a number of reasons. It's also important to understand what the comparative risk is in terms of human exposure, for example. So compost will always have traces of things in it, biosolids will, but since many of these things are coming from our uses in daily life, we have to think about where the human exposure is. So this is TCS, which is triclosan, which has now been phased out, fortunately, but it was antimicrobial that was added to toothpaste, and our exposure was much higher there than whatever happened in biosolids or compost. Plant uptake has also been looked at, and that's a key factor and a key concern. But again, most of these micropollutants or CECs do not seem to be taken up at significant levels in plants. What's key to sort of screening whether or not a chemical is of concern in compost or biosolids or other organics is whether or not they stick to, they're usually there because they stick to the organic matter. That's how they got there in biosolids, for example. They, rather than going out with the effluent water, they stuck to the solids. So there's a binding, an absorption that happens that can retain them in the soil. Many of them are broken down either in the treatment process, in the composting process, or in the soil subsequently. So the ones that are of greatest concern, though, as pointed out in several studies, is those that are, as listed here, have a high KOW, tends to partition to the organics. High toxicity, so obviously we're concerned about those things that are most toxic, have long half-lives, are persistent, they won't break down in the soil over time, and then if they bioaccumulate, they may affect some organisms more than others. So the ones that are continuing to be researched, and I'm sure we'll continue to hear about these and others as more research continues, are some of these. And as I said, Deb will discuss plastics later on. Risk assessment is part of the process here to understand, again, the presence of a CEC does not necessarily mean significant risk. So you've got to prioritize what you study and how it's looked at. Obviously, research has focused on the high production chemicals, so we're not studying things that don't occur very much in our daily lives or in society because likely there's just not very much of it around. But high production chemicals get a priority for research. The ones that are most toxic and the most persistent are the ones. The challenge is that going after chemical by chemical is pretty costly, and it doesn't sort of get at the potential synergistic concern. So an alternative approach, which I always mention briefly, is bioassays in particular as a screening tool. So bioassays are a look at the actual organism responses to a particular situation. So in this research, for example, from Ryerson University, where biosolids were land applied, they actually used a number of different organisms, both from the plant world as well as the animal world and the aquatic world, to screen whether there were not only lethal implications, was anything being killed by the biosolids land application process, but what were there actually changes in behavior and sublethal impacts. And pretty much the findings were that even using biosolids, and again, composts would probably have even less impact. The findings showed that biosolids had little negative impact on terrestrial biota. In general, no impact was observed. So that's an interesting way of looking at the research. The last point I'll make about CECs in general is the idea that biosolids, that you need to understand the context. And again, thinking of that triclosan example where our exposure was much greater in the actual toothpaste, we can look at this study out of, or compilation of information from the Northwest Biosolids about how many years of biosolids use it would be for the farmer to be exposed to the same amount as taking one tablet of ibuprofen. So the risk calculations done is that there's so little ibuprofen in biosolids, even though it's there and it's measurable, it's in such small amounts that a farmer working the fields applying biosolids every year it would take them 24,500 years to get the same amount of exposure as in 1 tablet of ibuprofen. So you kind of get what this chart is going after. It's not your exposure, our exposure is not through biosolids or compost, even when we're using them, handling them in home gardens or whatever. Our exposure is usually through the ways we're using these CECs in our daily lives. So PFAS, we'll talk about that now. I think it's an extreme worst case CEC, and it is the only common trace contaminant of drinking water that's being regulated in the low parts per trillion, including in Vermont. The reason for this is that there's widespread contamination by PFAS that's been in use since the 1950s. I won't get into the details. I'm assuming you know a little about PFAS or can look that up. But it's been used since the 1950s, various PFAS compounds, and they're found worldwide now in various organisms and matrices around the globe. You add that to that, the potential health concerns that have been uncovered over the last 20 to 30 years. There are hundreds of U.S. drinking water and groundwater PFAS contamination sites from industry and firefighting around the country. They're kind of scary because they don't break down in the environment, very persistent, because of the strong fluorine-carbon bond. And there's a great deal of public concern, and researchers and community groups are calling for action. And many states, especially in our region, are taking action. There's voluntary phase-outs being done over time. The most common PFOA and PFOS have been phased out voluntarily for use in manufacturing in the U.S. Things like ski waxes are being phased out now. Vermont has been a leader in that. So Vermont is aggressively addressing PFAS contamination, and so we'll look into what it all means. PFAS are in wastewater, biosolids, and compost because these materials mirror our modern life. So if you have food waste compost and the food packaging, which contains PFAS, has been in contact with the food, and some of the food waste has PFAS in it, and sometimes you're composting some of the food packaging, food containers, and those have PFAS in them. So we're aware of them now because of advances in environmental analytical chemistry, but we're measuring them also at very low levels. 1 part per trillion is equal to 1 second in 32,000 years. Very small amounts, and why the concern now is because they're being regulated, PFAS are being regulated at very close to background levels, including in Vermont. Leaching from biosolids and even food waste compost may not be able to meet these very low groundwater standards. This is what we're trying to wrestle with, is how to understand what the potential is for the materials that we're using and we're managing at facilities, whether or not we can ensure groundwater quality is protected at the levels that are being regulated. Vermont adopted last year the groundwater quality standards, and this year the MCLs, or drinking water standards, of 20 parts per trillion for the sum of 5 PFAS chemicals. And what's interesting is to look at, for example, a study on Cape Cod showing septic systems putting out PFAS just from our home daily uses of products with these materials in them, like our grain gear and our, as I said, waxes and floor cleaners and things like that. Those discharges from septic systems in this one study, where there was no industrial sources known nearby, impacted drinking water wells of neighbors to the tune of, here are the PFAS chemicals on the left here. This is the concentration in a logarithmic scale, so 10 parts per trillion PPT is here. And the groundwater and MCL drinking water standards for Vermont are quite close. So again, we're seeing PFAS in sort of general, neighborhood situations of groundwater in the single digits up into the low teens parts per trillion, which gets it pretty close to the Vermont standards. There are many uncertainties still about PFAS. The research has been ongoing and continues and will blossom for many years to come. Health implications are still being debated. Differences between different PFAS are important. There's this thing called precursors that some bigger compounds break down to shorter chain, more concerning ones. And Analytical methods are still in development, including for compost. There's no formal EPA method yet for that. Fate and transport in soils is being debated, and it is complex. So this has led to great variability in regulations. So here are the Vermont standards, but not that long before that, Canada adopted PFOA, and the two most prominent PFAS numbers for drinking water are 200 and 600, so an order of magnitude higher. So more research is needed. There's two major sources of PFAS. This is key. They're industrial discharges and firefighting, and 99% of the PFAS contamination sites identified across the country are from one of these two sources: in major industry use of PFAS and direct discharges to the environment, and occasionally through wastewater treatment, and then firefighting. This is the kind of data that we see as an example from Michigan, a factory that used PFAS coating on footwear, boots and such, and discharged to the environment over decades. And you know, we see groundwater in the 76,000 parts per trillion. Again, compare that to Vermont's groundwater standard of 20 parts per trillion. These are the kinds of sites that deserve attention, need to be cleaned up, and they are getting that kind of attention. The highest, North Bennington, Vermont, was the biggest prominent contamination situation with industrial impact. It was quite a bit lower in terms of its total numbers. The highest concentration in one set of Vermont data showed 2,330 parts per trillion, and yet that's still quite a bit higher than we see any connected to any facilities associated with biosolids or composting, for example. So this is kind of one of the worst case situations in Vermont related to biosolids management, biosolids and septic management, I should say. And here we're getting some groundwater numbers directly under a long-term site based on DEC data that shows up to 176 parts per trillion maximum, so considerably lower than the North Bennington site, and much lower than any of the major industrial. But these are still concerning because they are above the 20 parts per trillion. So these sites, again, this is a worst case kind of scenario, the worst known in Vermont at this point, and really one of the worst in New England that we've seen for data. And yet we're still not. It's important to keep the context that we're talking about, yes, elevated levels, but nothing like the industrial sites that we see elsewhere. The key thing is that what we're dealing with in compost and biosolids is ambient levels of PFAS, and those are these are more like background, right? Wastewater and biosolids and residuals, compost, paper mill residuals, these tend to have sort of ambient low levels, typically testing in the low single parts per billion of PFAS. Up to low tens of parts per billion of PFAS, compared to much higher levels in the industrial waste. So Andrew will provide more data on these specifics. But I'll just mention that we need to also consider what background is. So Vermont DEC sponsored a survey, which is very useful, being cited by many people around the region and elsewhere, regarding background soil levels of PFAS. And these are at sites that were not necessarily or known to be near any industrial or firefighting or other source. And yet PFOS, for example, was found in every single sample, PFOS being one of the two most prominent ones that have been phased out of use. But PFOS tends to linger in the soil, lasts for a long time. So it was found at every location, and then there were different levels. So these levels shown here are in nanograms per kilogram, which is parts per trillion. We usually talk about levels in soils and compost in parts per billion. So this would be, say, 5 parts per billion, 2 to 5 parts per billion. So we're seeing them in single digit levels of parts per billion, even in places where there's no significant known source for the PFAS. So these are unfortunately ubiquitous, and how we deal with them will be an ongoing saga. So thank you, and I will now turn this over to Andrew, and welcome, Andrew.
All right, well, Andrew's getting his slides ready. Let me give just a brief introduction. Andrew Carpenter is a certified soil scientist, certified crop advisor, and certified nutrient management planning specialist. He has extensive experience in the research, planning, and handling the technical issues related to the reuse of organic residuals. He earned a master's in plant, soil, and environmental science at the University of Maine, and he founded Northern Tilth, an environmental consulting firm focusing on organic waste management and building soil health in 2003. Andrew?
Thanks. So I want to start just by talking about the fact that as we get better and better about diverting organic waste from the solid waste stream, we're going to have more contamination, both physical and chemical. There's really no way around it. And a good way to think of that is sort of the comparison of a voluntary curbside collection program of organics versus a mandatory curbside collection of organics. So as you can imagine, probably most of the people on this webinar are like me. If they had a voluntary curbside collection program, they'd be very diligent about making sure that there was no physical contamination in what they put out to be collected. Once you go to mandatory, you get everybody, not just the tree huggers like us, but everybody is putting their material out, and a lot of people aren't going to be, just by nature, are not going to be quite as diligent about making sure they don't put any contamination in. So if California offers a great sort of example for all of us, because they are very aggressive in their recycling goals and have a lot of curbside mandatory recycling. We can learn a lot from them in terms of contamination. I also just wanted to say philosophically, as a company, our goal, really our mission, is to facilitate the recycling of organic byproducts back to the soil to build soil health. And by nature, it's a messy proposition. All of the materials that we deal with, whether they're materials that are can only be used by conventional farmers, or they're materials that can be used by organic farmers, they're all imperfect. They're not synthesized in a laboratory. They all have the potential for some of this contamination. So really, to be part of this, you really have to have tolerance for the idea that there is going to be both physical and chemical contamination of materials that we're returning to the soil. If we go to PFAS, one of the things I want to mention, and Ned made, I think, reference to it, is that it's so ubiquitous and it's been used so widely in a lot of food packaging and things like that, that we ourselves have had higher concentrations in our blood than we do now, because PFAS and PFOA have been phased out. But it's dropped almost, I think, if you look back far enough, almost an order of magnitude in the general population in our blood. Interestingly, for some compounds, our blood level is high enough that if we were a biosolids, we couldn't be land applied with the current restrictions that are in place in Maine right now for PFAS itself. You often see things quantified, some of these ubiquitous compounds in household dust or in daycare center dust, just because it is so widespread. If you look at PFAS, it's in so many things, so many household products, that our dust happens to have very high levels of it. Tends to be higher than you find in biosolids or compost, biosolids-based compost, food waste compost, or that kind of thing. And we've dealt with this before. Polybrominated diphenyl ethers, which are flame retardants that basically been used in almost everything in our household you can imagine. They tend to be high there as well. Our exposures to all of these things tend to be a lot higher in these consumer products or in our household than the potential for it to go to soil and then to plant, or from soil to water and then us be exposed to it that way. It's not to say we can downplay it and say it's fine, but it is sort of the nature of, as a society, really happy with the convenience that a lot of these compounds bring us. One thing I think is interesting is the fact that in concealer or foundation cosmetics, it's a couple orders of magnitude in concentration higher than you'll find typically in biosolids or other composts. One of the things is, and Ned made reference to it, biosolids, which I work with biosolids, I work with all kinds of residuals. Biosolids tend to be the ones that get more scrutiny, and in general, compared to the other residuals I work with, or byproducts, they also tend to be higher in some of these compounds. It's not always the case, and typically if they're in biosolids, they're an indication that they're probably in other things as well, because they are, biosolids tend to be somewhat of a mirror of what we use in society. So in Minnesota, they looked at surface water from non-biosolids composting sites, from food waste composting sites or food waste and yard waste mix. And in their study, they found that every one of the sites that they looked at surface water at, they had at least one PFAS compound that was above their action standard. Now it's probably a reflection of their action standard being fairly low, but it is the case that even non-biosolids related materials do have enough in it that you can see it getting around, a little bit mobile. One of the things that's tricky about PFAS is the physiological characteristics of it make it particularly tough. Ned was talking about the absorption of these compounds to solids. Typically, it's been the case that the more recalcitrant or longer-lived organic compounds also tend to be absorbed strongly to solids, so they're not soluble. Problem with PFAS is it's both long-lived. It does absorb to some extent, but it's also leachable. It's also soluble, and so that makes it a particularly hard one to deal with. This is a study that came out of Purdue University a year and a half or so ago, and it's looking at PFAS compounds in compost, and these are all non-biosolids compost. The 7 of them, 1 through 7, all, well, actually, with the exception of the first 2, all contain primarily food waste and leaf and yard waste, green waste. The last 3, I think, are only yard waste. But the thing that's interesting about this is if you look at just the PFOA level, the screening level for biosolids in Maine, but the DEP is 2.5 parts per billion, and almost all of these composts would not meet that screening standard, even though they wouldn't be required to be screened in Maine because they're not biosolids, but if they were, currently they wouldn't meet that screening standard. This is, again, not to say they're terribly contaminated. These are actually fairly low levels. More the fact that they can't meet the standard is more of an artifact of the low standards that we have in Maine. But they're ubiquitous, so we're going to find them in food waste compost. There's some conjecture at the end of this article that it may have come from compostable foodware. That's possible. It's also possible that it's something else. I wouldn't necessarily point the finger at foodware. I think that the thing that's interesting about PFAS is it's hard to point the finger at anything, and if you do, you're likely to get in trouble because it's really coming from so many different sources that it's pretty hard to pinpoint. This is I bring this up because this is in Maine now, and this may end up happening in more and more states as we find that there are stricter and stricter regulations related to PFAS for biosolids compost, and septic-based compost. For land application sites, we're now required to not only test the material, but test the fields that they're applied to. And based on the amount that's already in the soil, we have cumulative loading rates. So we're basing our loading rates of these materials on how much PFAS they have and how many years it would take at the current levels of PFAS, primarily PFOA and PFOS, to reach the screening standards we have. And again, it's 2.5 parts per billion for PFOA and 5.2 parts per billion for PFOS is the screening standard that they have in Maine currently. So this is just an example of one septage compost and the going through the calculations of if we're applying this at typical agricultural rates, how much are we increasing that concentration each year, and essentially how long can we do it? Just to give a little bit of the scale, the Vermont background soil levels from uncontaminated sites are on the right, 0.52 parts per billion for PFOA and 1.1 for PFAS. At this rate, this agricultural application rate with this septage-based compost, we're adding, essentially increasing the soil concentration 0.04 parts per billion per year. It's also the sampling that goes along with PFAS. It's interesting because it sort of gives an indication of how widespread it is in our society. We have to take huge precautions when we're going out sampling, including not using shampoos, not using rain gear, so certain types of rain gear when we go out. There are all kinds of personal care products that you can't use before you go out sampling for PFAS, because we're looking at it in such low concentrations, and they tend to be there in significant concentrations in things we use. We also have to do a lot of decontamination in the process. To me, this is interesting just in the fact that testing at these very low levels, essentially we're having to make sure that our bodies themselves are not getting the same levels that they typically get when we go out to sample, so that we ourselves don't contaminate the sample. Speaking of recalcitrant compounds, there are some very resistant herbicides. When I say resistant to composting, so they're also recalcitrant, maybe not quite so much so as PFAS, but some of them are pretty damn resistant to composting, and aminopyrrolid is one. It's used very widely. It's a very effective pesticide. One of the reasons it's so sought after is because such low concentrations are really effective in killing certain, broadleaf plants. And it will not degrade in the composting process. So if a compost facility happens to be getting, say, grass clippings, or even in some cases manure from horses that are feeding on pastures that have used some of these pyrrolid-based pesticides, it shows up in the compost itself. Because it's not broken down and you're actually breaking down organic matter in the composting process, it can actually get a little bit concentrated in the compost. And in the end, you have a compost that, even though the levels are so low, very hard to actually detect, by common analytical methods, it can still be effective as a pesticide. And this is especially true with aminopyrrolid and certain legumes like the fava beans that are shown here. And again, it's an artifact of this, that we use, the fact that we use these pesticides relatively widely. You can find it in compost that are certified for organic use and other compost as well. But it's not coming from direct injection of the pesticide to the compost, but just a small residual coming into the facility. This thing on pyrrolid a little bit more recent, but pyrrolid, we've had outbreaks of that in compost causing problems with plant growth for the last decade and a half. There are some effective uses, remediation tools for this. One is char-like materials, materials that have sort of a graphite-like structure of the carbon, essentially some graphite or char in them, add a lot of absorption and are able to absorb some of these recalcitrant pesticides and make them ineffective. There are some compost facilities that actually use char-like material in a prophylactic way, just in case they do get any pesticides in the process. So there is, there's something that can be done about it. There's also people that are working on trying to take these pesticides out of use or making sure they're not making their way to compost facilities. It's a tough task. I want to talk about physical contaminants. We've done a fair amount of work on the West Coast where this is is a bigger issue than it is here. And again, I go back to say the difference between people like us separating out our our organic waste very diligently and having an entire population all of a sudden go to mandatory composting. And also just there are different processes out there now for extracting the solid, the organic portion of solid waste from the waste stream. Some of them actually happen after the material is collected. When you do that, you're bound to have more physical contamination. And primarily in the states where this is sort of more advanced are film plastic, hard plastic, metal, glass, and then you'll find other identifiables. But again, I think the take-home message, or the message I want to drive home is that the better we get at pulling organics out of the waste stream, the more physically contaminated, or the more physical contamination we have to deal with. And since I'm running a little late on time, I won't go through this, but I just want to point out that California and Ontario, and there are other places, other regions that are coming up with actually developed standards not only for quantifying the amount of contaminants that are in compost, but also standards on that compost. And I can say from my experience that the one that can be tough is getting down to 0.1% of film plastic in the final compost. So there are methods out there for dealing with these, and with physical contamination. One is, very simple. It's pick station. One on the upper left is the site visit. It takes material from the mandatory curbside collection program in San Francisco. It's essentially a belt with hand pickers on it, and then the one to the lower right, it's a facility. It's a little smaller, maybe 60,000 tons per year. So from the Portland, Oregon area, it's got a little bit smaller system, but the same. That's simply prior to composting, having hand pickers. There's one thing I did want to mention that the facility down to the right pointed out to me is the problem that they have with, compostable bags are great in terms of increasing diversion rates, but they can be pretty tough on composting, and it's on the front end. It's almost impossible for them to identify the compostable bags from other bags that they're getting in the process. So that can be a challenge. Not to say that compostable bags is not good. It's great from a collections end. Sometimes it can be a little complex on the, sorting out the physical contamination end, especially on the front end. There are also depackagers now, which it's a great advance. It allows you essentially to throw boxes of spent food right into these systems and separate the plastic, cardboard, stuff like that from the material that you want to compost or digest. This may have some of the portion that you don't want, and the organics has the potential to be broken down here into smaller particles that then ends up in the compost or the feedstock for the digester. Another thing is that this facility does in the Northwest, I mentioned, that I think is great. They worked very hard at this, but they came up with a system for actually charging the people that are bringing food waste of their material by the amount of physical contaminants that they pick out of the feedstock. So they went through a very detailed analysis of how much it costs them, and then they in turn shift that to the hauler, and of course that gives incentive for the hauler or whoever's providing it to the hauler to get the contaminants out before it comes to the facility. And then there are post-processing methods, and one of them is, which I've seen a couple of out again on the West Coast, is during the screening process, when the material's getting thrown in the air a little bit, using vacuums at that point to get a light fraction out, and that is primarily film plastic, and they're fairly effective. So if you look at this, materials being screened here, this is a vacuum here, and it's pulling it into these bags that they then end up taking to a landfill. If you look at this slide, just look on the far right here. This is 0.5% of film plastic in a kilogram of material. Like I say, I mean, it looks like a lot here, but with the at 0.1%, that can get pretty low, especially for feedstocks that have a fair amount of plastics coming into them in the first place. A couple methods that TMEC has come up with for identifying physical contaminants. One is the drying sieving method that most people use. Wet sieving I've used on some projects where there's greater contamination. It seems to be a little bit more effective, but I won't go into that. I'm actually at the end. So also wanted to put a plug in for this video that we put together for the Composting Council Research and Education Foundation. They're sort of a wing of the U.S. Composting Council. This is the YouTube address for it. It's a two-part series, somewhat relevant to what we're talking about here, but it's instructions for collecting good representative samples, and also talks about the potential contamination issues and sanitation methods that you want to take into account when you're doing sampling. So in summary, really, I guess, want to get across the fact that, you know, especially with Vermont's more aggressive goals, as we get are successful at getting a bigger portion of the organic waste out of the solid waste stream, we will run into, it's almost inevitable, more physical and chemical contamination. It means a couple things. We have to have tolerance. We can't assume that there's not, you know, things that we're not testing for are just not in there because it's, say it's an organic facility or something like that. Risk assessment, the type of thing that Ned talked about, it helps us in trying to determine, so yeah, we know this is ubiquitous in society, so how much of it is bad and how much can we tolerate? And there are options on the collection side, on the pre-processing side, post-processing side, and even as far as the persistent herbicide goes in terms of blends, they can help deal with some of the contamination that we get.
All right, I'll just finish up with just a brief presentation about microplastics. Andrew alluded to these as he was looking at some of the devices for separating the packaging material. But just to clarify, this term microplastic is really referring to the size of these particles, not the chemistry of the plastic. Okay, so, and these can be defined in a couple different ways. Primary microplastics would be those that begin their life as very small particles, defined as less than or equal to 5 millimeters in size. And really, our world is full of microplastics. And I just name a few of the items that contribute to microplastics, you know, including the clothes that we wear, and a lot of the, you know, personal care products that we've used also contain them, and some of the industry and technologies. The secondary type are those that are fragmented from larger plastic products. So this could be, you know, water, soda bottles, for example, or plastic bags that break into smaller pieces. In the lower right-hand panel there's some little arrows by pieces that are denoted to be secondary microplastics, often recognized because the edges tend to be jagged, and that suggests that they are broken down from the primary. So as Andrew mentioned, you know, there's been historically some concern about the contribution of depackagers, but I'll have to say, you know, there's been some evolution in technology to try to address this as well. Early models that used hammer mills tend to splinter the packaging, which allowed some of these particles to pass through the screen and then go into the environment. This was recognized, and so technological advances have been trying to reduce the amount of the applied force to reduce that amount of splintering of the packaging. And just illustrated is a depackager that's at E.L. Harvey and Sons in Westboro, Massachusetts. So you can see the type of technology used there. And that's a facility that has been taking in food waste. So why worry about microplastics? The biggest concern has been the potential for bioaccumulation in the food chain. I think what usually grabs our attention are these like we see pictures of islands of plastic floating in the oceans and so forth. But this is also a concern that's we're also recognizing on land in terrestrial systems as well. The big issue is really that microscopic plants and animals think the way that those microplastics kind of glitter and reflect, they think they're food and they consume them, and it's basically representing empty calories. And then as you move up the food chain, organisms start to accumulate larger and larger quantities of those in their bodies. And some recent studies have been showing, even earthworms have evidence that they're being exposed to microplastics. In the soil food web, they're actually up the food chain about 4 or 5 positions. Well, maybe sometime, I guess I should say between 2 and 4 trophic groups above the bacteria that might be, and fungi consuming them. But we can see it as we look, microplastics are on the epidermis of the earthworm, and we can tell they're going through their guts because we can see them in the middens and in the casting materials. A recent study, I'd say a lot of really top work on terrestrial ecosystems is coming out of Germany, and their scientist, Matthias Rehlig, and his colleague Lehmann just came out with this news-breaking article in Science magazine a couple months ago, and making some hypotheses about how these microplastics may be moving through our food chain. Unfortunately, compost doesn't really break down microplastics, and that's been one of the concerns, but there are people scouting for potential organisms that could metabolize microplastics. And a couple examples that have emerged in the literature within the last couple of years are a group of scientists out of Stanford University identified beetle larvae, otherwise known as mealworms, actually can metabolize polystyrene, and with a byproduct of carbon dioxide. The fact that the final product is that tells us that they are metabolizing them, and it would be it's kind of the gut microbiome that's responsible for that degradation. So that's been investigated. And then within the last year or two, we've a group of scientists out of Japan kind of came upon a bacteria that can break down the plastic found in water bottles. And so these are I just share these with, the idea that there is promise for identifying microbes that could be used in remediation. And with that, I'm going to kind of conclude and leave this contact information for all of the speakers today, so that if you'd like to follow up with this with specific questions and continue the discussion, you can, but I'll turn it back to Natasha to host the questions.
Great, thank you so much to all of our presenters, Deb, Ned, and Andrew. Those were great presentations. Deb, one of the questions that came in for you was about the slide where you showed the effectiveness of composting on reducing the abundance of antibiotic resistance genes. And there was a question, the other one that was right up there with compost was the drying, dewatering being quite effective in that slide. And so the question is, if you have a sense of how that works, or if drying or dewatering could be combined with composting for a more effective reduction.
That's a great question. I don't have the best answer. The one thing I did notice, and why I didn't emphasize it, was there was only 4 studies, and N equals 4 of the drying, dewatering. So I just was a little concerned that I wouldn't want to extrapolate it too broadly. So that was, about 10% of the number of studies that we saw for composting. So I'm struggling with understanding exactly what would be the mechanism that would account for that activity. So I did notice that it was statistically significant. So it's the sample size was the main reason I was hesitant to jump right out there and adopt that. And I think you would want to have additional research to know whether combining those would be a good practice or not. This depends on why are you drying and dewatering. I've typically seen that more with the anaerobic digestion and some of those products, which were less effective in the the, in managing those.
Yeah, thank you. And that's a good point. I wonder if that would even potentially be a more natural partnership of methods with some of the digestion techniques which were lower in effectiveness.
And I'll just add another factor that sometimes people get concerned about recolonization or something of pathogens. And one thing that we have to keep in mind is one of the things that we look at as a measure of proper composting is whether that material is stable at the end of the process. And sometimes drying can be a mask of stability. And if it's too dry, it might give you the illusion that it's a stable product. But if you add moisture again, it might start to become active again. So I'm a little leery of just relying strictly on drying, especially because a lot of these microbes have ability to go dormant under those stages.
Mm-hmm.
So I approach it with caution.
Thank you. And Ned, we did have a question in that you did reply to via chat, but I was wondering if you might just quickly address it for all of the participants who may not be reading along the chat line in terms of how PPCPs are phased out and what could replace them to serve the same purpose. And it sort of gets at what I think all of you have brought out a little bit in terms of recognizing sort of the societal or cultural contribution, like this is why we have these contaminants, because they're in these products. Recognizing that as a main contributing factor, how do we sort of rearrange ourselves as we try to address this and not potentially replace something that's been identified as a problem with something that may be even more problematic?
Yeah, and it's a good question that Elena brought up. We can't phase out some medicines and life-saving chemicals that are in use. So nothing's going to be perfect. I think that's the bottom line. We have to weigh and balance things out. There are steps we can take, however, and research and understanding, again, whether something is present does not necessarily mean there's any potential risk. So research and understanding for these different trace CECs is important. I'll emphasize again that most of them are broken down in composting or in the soil. Numerous research has shown that. So it's really the persistent ones that might have impacts on biology or on humans that we have to be concerned about. And what's happening there is that there is some efforts in green chemistry. Essentially, if there are certain pharmaceuticals, for example, being developed that end up being very persistent at this point, green chemistry is the practice of trying to find ways to make those molecules automatically break down over a period of time after they've had their effect in the human body. And that way, basically designing pharmaceuticals more for the full life cycle to ensure they're breaking down. So that's one promising thing, not very far along, but certainly something is beginning to seep into consciousness. And so I'll leave it at that. Again, I think we have to understand that there's going to be a balance, and we need to accept, as Andrew was noting, we need to be able to tolerate some contamination compost when the overarching benefits of recycling these materials, taking advantage of the nutrients, getting the organic matter back into soil, building healthy soils, et cetera, have so many benefits that we just need to control the risks as best we can.
Mm-hmm. Yeah, thank you for that. And I think that sort of leads into the next thing, and it's something that Andrew touched on and you just now, Ned, in terms of this larger picture and how we as a community or as a society balance we have these higher goals or increased goals for organic diversion from landfills. We still need to do something with it. And then as we bring people along, we're sort of introducing a wider variety of potential contaminants, both micro and macro, into our compost or digestion streams. and sort of seeing it as a larger system and a bigger picture, as opposed to really focusing in on you know, just this is the problem as the end result, but like how does that fit as a complex in our larger goals? One of the things that I've seen is, and I really appreciate this session and all of your presentations, is a real need for education, even amongst the organics community, in terms of educating ourselves to better educate policy makers and regulators. I've had some conversations where it seems that the background levels that Ned and Andrew, you both touched on, are almost ignored, and compost being singled out, whereas for like road projects, you know, fill might not be, there might not be limitations for that, but if you're going to bring compost in for revegetation, there are regulations around contaminants for the compost piece of it, even though the fill may be as contaminated, if not more so, just from background levels. And so I guess it's just an observation, and if any of you want to add to that, it's just how do we have these conversations and continue to educate everybody involved in the decision making and regulating that is part of our industry.
Yeah, I'd say I think two things related to that. One is a better understanding of these materials in the context of our daily use, because it is, I mean, coming from, because I've worked with biosolids for such a long time, and biosolids tend to be such an easy scapegoat for these compounds. It is, and once there's, you know, some public incident related to them, it's hard to back away from it and say, well, wait a minute. I mean, yes, we have polybrominated diphenyl ether in the biosolids, but look at, I often think of like, I tend to buy older cars where the seats are starting to fall apart. I know there's chock full of polybrominated diphenyl ethers. I also eat junk food, so I've got French fries that have spilled onto the seats, and then I eat them. And the polybrominated diphenyl ethers are lipophilic. They love the French fry. So I'm just eating them directly. And that, you know, it's a much bigger exposure than we're ever going to have from using that material again in low concentration, much lower than in my feet, as a soil amendment, and then growing something. And it does vary by compound. Polybrominated diphenyl ethers like dioxin tend to be, you know, lipophilic, and they absorb to soil very strongly, so they're not soluble, they're not taken up by plants. PFAS is a little bit different, but I really think like looking at it in the larger context is really important. But the other thing is, having dealt with biosolids for a long time, it has made me more sort of maybe pissed off by our lack of questioning about all the things that are in our daily consumer products. And to me, you know, a great example, people could disagree with me, and I might be wrong. I don't think I'm wrong, but anyway, triclosan and triclocarbon have been used for a long time. They're not as used as often anymore. They're antimicrobials. They're in toothpaste, hand washing, all sorts of things. And it's been clinically proven that they are not more effective than soap and water at reducing your exposure to pathogens. And yet they were used, you know, I don't know how much on a daily basis, but huge quantities of them. They didn't really have any effect, but they were marketed really well. And then, you know, those people dealing with the byproducts on the other end, you know, mostly biosolids, but other things as well, are left, you know, dealing with the negative sort of public fallout from the fact that we have the residual levels of these things. So I feel like we could do a better job, you know, starting up the pipe a little bit. On our end, we tend to be reactive and don't have time to sort of go back up the pipe. But you know, as a society, it would be great to see that.
Yeah, and even in food itself, I think the Purdue study pointed to food we eat as a PFAS source as well. And so,
yeah, absolutely.
Everything that we put in our bodies and use on our bodies contributes to the issues.
I'll add that there's hope, though, and there's positive things. Two things. One is our life expectancies have continued to rise over the years. So whatever we're doing, we're still healthier than we were in the past. So that's good. It doesn't mean an excuse to ignore the issues, but giving, every now and then step back and look at the big picture, and we're pretty well off. The other thing is, as composters, as those dealing with waste streams, we're in a unique position to help society recognize when there are threats, because we're looking at the back end, and if there's something that really can't be recycled, if there's something coming through that is dangerous, we often the ones, the sentinels who notice it because it's impacting our compost. Like the clopyralid issue is a great example. It's like, wait a minute, this is something we need to do something about. We recognize it here at the tail end, and society, we need to correct here. So trying to recycle all these things, food waste, biosolids, anything, is a noble task because it helps society understand where we've gone too far in terms of creating things like PFAS, some of them of which should definitely be phased out. So we're we're the sentinels helping society correct.
I wanted to add on to this conversation, and that was just a pitch for kind of the idea of maybe some standards and or labeling issues with this in terms of, I think we have to think what kind of material and what kind of exposure it could have. Like you mentioned, the landfill or that may be able to handle a little bit higher levels of materials that might be ingested or brought in through respiratory system. Those tend to be more toxic than those that you can have a little bit of dermal protection against. But I think, as far as if a consumer is going to be purchasing compost for use, I think it's fair for them to also know what levels of certain contaminants may be present in these materials, because you may choose to use them in different ways depending on what those concentrations and materials are.
I both agree and disagree a little bit, just based on the fact that, say in Maine right now, we actually are required for composts that are biosolids and septic based, but not for other materials. So it makes it appear, if it's not across the board, that those two materials are the ones that have it and other materials don't necessarily, which we know not to be the case.
Yeah, I mean, that's a good point. I'm just thinking the issues with compost are kind of like where we were with certified organic, maybe 20 years ago. We throw a lot of, and you were pointing out some of the inconsistencies in these thresholds for different states, different countries, et cetera. There's not much uniformity out there.
Yeah, right. I agree. Yeah.
Great. Well, thank you all for those closing thoughts and discussion. We are right at 2:30, so I think we're going to end the webinar here, and we hope that you will join us for some of the other webinars that we have later on this week. Thanks, everybody.
Thank you, Natasha, for organizing.
Yeah, thanks a lot.
Thanks for joining, everybody in the audience.